Welcome to this lecture on expression vectors in animal biotechnology. Today, we are going to explore one of the most powerful tools in modern science. We will learn how scientists hijack the cellular machinery of microscopic organisms to mass-produce life-saving medicines. By the end of this session, you will understand both the biology behind these microscopic factories and the mathematics used to scale them up for global production. Let us start with a simple analogy to build our intuition. Imagine you have a highly detailed blueprint for an advanced sports car. If you put this blueprint inside a standard, highly secure storage vault, it is perfectly safe. You can make as many photocopies of it as you want, but you will never actually get a physical car. A standard cloning vector is exactly like that storage vault. However, if you take that same blueprint and hand it to a fully equipped manufacturing factory, the workers will build the actual car. An expression vector is that manufacturing factory for our DNA. It does not just store the genetic blueprint, it actively commands the host cell to manufacture the corresponding physical protein. Now that we understand the factory analogy, let us look at the formal mechanics. In molecular biology, an expression vector is a specialized plasmid or viral vector engineered specifically to facilitate the transcription and translation of a cloned gene. While standard cloning vectors only need an origin of replication and a selectable marker, expression vectors must be equipped with highly active regulatory sequences. These are the factory managers. They include a strong promoter to initiate transcription, a ribosome binding site to start translation, and a transcription termination sequence to properly end the process. By inserting our gene of interest downstream of these elements, we effectively hijack the host cell's machinery. Before we move on to real-world applications, let us see this with a concrete visual example. Here is a schematic diagram of a typical expression vector. Notice the circular structure representing the plasmid DNA. On the top right, the gray region is the origin of replication, which allows the plasmid to copy itself. The red section is the selectable marker, usually an antibiotic resistance gene, ensuring only cells with our vector survive. But the true power lies in the expression machinery, the green promoter region and the yellow ribosome binding site. These sit right before the blue multiple cloning site, where we insert our gene of interest. Finally, the orange transcription terminator ensures the protein manufacturing stops at exactly the right point. Having seen the structure of these vectors, we must ask, why does this matter? The development of expression vectors completely revolutionized modern medicine. Before their invention, obtaining therapeutic proteins like human insulin required extracting tiny amounts from the pancreases of thousands of slaughtered pigs or cows. This was incredibly expensive and often caused severe allergic reactions in diabetic patients. Today, the process is entirely different. We simply insert the human insulin gene into an expression vector, put it into bacteria or yeast, and grow massive vats of pure, safe human insulin. Beyond insulin, these microscopic factories produce life-saving vaccines, human growth hormones, and even industrial enzymes used in food processing. Without expression vectors, the entire biopharmaceutical industry would not exist. Now that we appreciate their real-world impact, let us understand how biotechnologists measure their success. To predict how effective our expression vector is, we use a basic mathematical model to estimate the total protein yield. The formula is Y equals N times C times E times M. Let us break this down. Y is the total yield of our recombinant protein in grams. N stands for the total number of host cells in our culture. C is the plasmid copy number, meaning how many vectors reside inside a single cell. E denotes the expression efficiency, which is the number of protein molecules produced by a single vector. Finally, M is the molecular mass of the protein, which converts our molecule count into a measurable physical mass. If we engineer a stronger promoter, E increases, which directly boosts our final yield Y. This formula we just saw leads us to a practical example. A student is growing E. coli containing an expression vector for human growth hormone. We are given 2 × 10⁻10 cells. Each cell has 50 vector copies, and each vector produces 1,000 molecules. The mass of one molecule is 3.6 × 10⁻20 grams. We need to find the total yield. We substitute our known values into the yield equation. First, we multiply the cells by the vectors per cell, giving us 10⁻12 total vectors. Next, we multiply by 1,000 to find that 10⁻15 protein molecules are synthesized. Finally, multiplying by the mass of a single molecule gives us our final answer, 3.6 × 10⁻5 grams, or 36 micrograms. This proves even a small culture produces a medically relevant dose. Now that we have used the basic yield formula, let us look at how industrial biochemical engineers approach this. In a real-world commercial scenario, a pharmaceutical company usually knows exactly how much drug they need to produce. So, we must work backward from a target yield to find the required biological workforce. The formula is N total equals Y divided by P. Here, N total is the total number of host cells we need to grow. Y is our target yield in grams. P represents the production capacity, which is the tiny fraction of a gram of protein that one single cell can produce. By dividing the massive target yield by the microscopic production of one cell, we determine exactly how many bacterial factories we need to hire for the job. Before we move on to sizing the bioreactor, let us see this workforce calculation with a concrete example. A pharmaceutical company needs to produce exactly 5 grams of recombinant insulin for a clinical trial. They know their optimized expression vector allows each cell to produce a total protein mass of 2.5 × 10⁻13 grams. We set up our equation, N total equals Y divided by P. We substitute 5 grams for the numerator and 2.5 × 10⁻13 for the denominator. Performing this division gives us a staggering final answer, 2 × 10⁻13 individual cells. This massive number represents the absolute biological workforce required to successfully meet our pharmaceutical production goal. So, we have proven that we need trillions of cells. This brings us to a critical engineering question. How much space do they need? We have to respect the maximum cell density limits of our bioreactor to ensure the cells survive and produce protein. To find this, we use the volume formula. V equals N total divided by D. In this equation, V represents the required culture volume in liters. N total is the massive cell count we just calculated. D is the maximum cell density, which tells us how many cells can safely live in a single liter of liquid. This crucial step translates our biological cell count into a physical engineering parameter, allowing plant managers to size their stainless steel tanks appropriately. Let us finish our commercial production example by calculating this volume. We know from our previous step that we need 2 × 10⁻13 cells. The industrial bioreactor supports a maximum cell density of 10⁻9 cells per liter. We use our volume formula, V equals N total divided by D. Substituting our values, we divide 2 times 10 to the power of 13 by 10 to the power of 9. The final answer reveals that we need exactly 20,000 liters of culture volume. This means the company will need a massive industrial-scale bioreactor to produce just 5 grams of pure insulin. It perfectly illustrates why biotechnologists constantly work to engineer stronger promoters to increase per-cell yield and cut manufacturing costs. We have covered a lot of ground today, so let us summarize the key takeaways. First, remember the fundamental difference. While cloning vectors merely store DNA, expression vectors act as active biological factories. They achieve this using essential regulatory sequences, like strong promoters and ribosome binding sites, to manufacture proteins. This very technology is the beating heart of the biopharmaceutical industry, enabling the mass production of life-saving medicines like insulin and vaccines. Finally, we saw how mathematical modeling bridges the gap between microscopic biology and macroscopic chemical engineering. By understanding and optimizing vector efficiency, scientists can scale up production from tiny laboratory flasks to massive industrial bioreactors, saving millions in manufacturing costs. Thank you for joining this lecture on expression vectors.